sir. Yes, sir. Thank you.
Okay, so so right now, I'm just going to discuss about this plant research for sustainable agriculture. And always, I talk about the way forward for the food security, because that is most important for all of us. So next slide, that food security, we all know that it is important for our country. So what I'm going to discuss today is mostly about introductory part of the food security, then food security itself, and how climate changes affect the food security, plant biotechnology, and food security, and new technologies and way forward. The last point I'll be elaborating more compared to the previous points. So that if you talk about the agriculture biotechnology, so right now we do have so many problems existing, like drought tolerance, water use efficiency, yield increase, the nutrient use, increased shelf life of various fruits and vegetables. The disease tolerance is also one of the most important, and my lab is working on this aspect. And the herbicide tolerance is also one of the key factor that right now people are trying to implement in Indian subcontinent also. And salt tolerance, because most of the many part of our land is now becoming the is because of urbanization. The land which was actually agricultural land is now being occupied by the urban development things. And now the cultivation and our farmlands are now shifting towards the periphery. So that they are also having new soil, new things are there. So salt tolerance is one of the most important thing. Then aquaculture and all these things will be, if we talk about in the context of the sustainable development, then we have to think about the land conservation, reduced emission, reduce waste, water conservation, which right now is very important aspect, and the carbon sequestration. So what I'm going to discuss about, many of these aspects in my lectures with the recent advancement in the plant biotechnology. However, I will be also discussing about some of the part which we have already got during our first green revolution. So I think you all know about the green revolution, which has happened in 60s, 70s, and 80s. And what we have achieved from those first green revolution, and that I'm going to discuss. And in the end of my seminar, I will be sparing near about 15 minutes, so that we can interact with each other, and you can ask me any type of questions related to the topic that I have covered. So, when we talk about the first green revolution, so this green revolution has started, long back in 60s, 70s, and 80s. If you talk to your grandparents, sometimes it was there, when our Prime Minister was Lal Bahadur Shastri. And that time, you all know that he has urged the masses, Indian masses, to do at least one day fast, so that we will have some food security when the USA has stopped. Hello, can you hear me?
Yes, sir.
Yes, sir. Yes,
Yes,
sir.
sir. It's audible.
Some noise was coming. So, that time, what has happened, like the USA has stopped, sending the wheat and everything. So we have started our own, green revolution. And you all know that, this person, Norman Borlaug, he is the father of the green revolution, as well as our, just now we have lost him, the MS Professor MS Swaminathan, who has been recently awarded as a Bharat Ratna. So the important thing, what I will just discuss about these things, like the Norman Borlaug has not got award in the field of science. He has got a Nobel Prize in peace. So if your stomach is filled, so there is a less chance of fighting with each other. So that's why it's very important to fill first the stomach of masses, and then only we can talk about the other things in for the new growing population, and what are the different difficulties and everything which is happening. So in this case, in the first green revolution, what they have done is that, if you this the data, if you see in this graph, these are the data of India and Pakistan, because that time the both places the green revolution has started. And here you can just see that the most of the yield per kg per hectare was very, very less. However, the Mexican wheat was very high yielding. So what they have done is that they have taken wheat and rice both. They were short in height, although they were disease susceptible, and they were high yielding. However, the Indian varieties were low yielding, but they were disease resistant. So what we wanted to retain in the breeding program is the character of the basic characteristics of the Indian varieties. And the variety which we are going to introduce is where the characters like the high yielding, as well as the short, the dwarf varieties that we just wanted to cross with each other. So what they have done is that after the crossing, they have the in the strategy what they have adopted during the green revolution is that they used the high yielding varieties. They also started using chemical fertilizers and pesticides. I'll come to this particular point because this particular workshop is of yours, in HRDC, is also related to the sustainable development. So we will discuss about these, the use of chemical fertilizers and pesticides in detail. And then the new projects were built, the irrigation potentials were generated, use of tractors, motor pump, and farm machineries, they got increased. And then, there was a nationalization of the banks during that period. And then the bank lent the agricultural loans to the farmers. So these are, the some of the development which has started with the breeding programs during the first green revolution. So there, say what is conventional plant breeding. If you'll understand these things, then probably many of you who are from the botany background or the biology background may be knowing these are the some of the basic things. But I am just giving this idea to other people who are not from the plant field. So when you'll have a recipient where, like in the wheat which was growing in India was recipient, and the donor was the Mexican wheat, that you can see there. So what you are going to do is that you are going to cross them. F1 generation will be, all is will be, Mendelian in a first law will be there. So it will be having only the dominant characters will be shown. But in F2 generation, there will be a segregation. And after the segregation, you go for the phenotypic selection. And after these phenotypic selections, you can go, do all these things manually. That I do have certain slides where you can have either go for the glasshouse trials or many times go for the field trials. And then you can release the variety if that is very important for the farmers. So there is one, the example which has started, the long back with the submergence tolerance rice, which can survive floods as long as near about 17 years back. So what they have done is that there was two varieties of rice. One variety of rice was susceptible to flooding. Another variety of the rice was which we know the gene. It is sub-1 rice, which is actually is growth arrest during the flooding, but enhancing after the survival. So after the 17, 18 days of the flooding, the susceptible variety will be, when water retreats, it will be dead. But the varieties which will be the sub 1 variety or submergence tolerant variety, that will be, they will survive. So this type of things, how they have done, the this breeding programs. This example I'm just giving you, this example, just to understand that what is, the basis of the breeding programs. So there, the Swarna variety was the, susceptible variety, and you can just, just concentrate on the orange color of the Swarna variety. However, the sub variety was the low ending, but it is having the variety which is having one gene or one locus which was, important for the sub 1 or submergence tolerance. So this I have kept in yellow, and other part of the recipient variety, this is, in gray. So when you cross them, so what will happen, like this the it will be just, almost 50%, 50%. But since, the sub 1 will be also integrated here. So hello.
Yes, sir. We can hear you, sir.
Oh, some noise is coming, so I got confused. Okay, no problem. So what happened, that if you say that, the variety which is having this yellow color, which is, the sub 1 trait is there. So then you go for back crossing. Back crossing you are going to, because you want to keep in most of the characters of Swarna, but only the sub 1 is that you want to integrate into Swarna. So you just go for the back crossing with the variety which is recipient all the times. So near about 7 to 10 generation of this back crossing, you will find, that almost 80-90% of the genome is just like Swarna, and it is having the sub 1 variety. So this I am just given you just for example at how this is going to help. So you can just see that, this Sarna sub 1 variety, which is now erect, and after the flooding, which is the susceptible variety, it just growing side by side, it is dead. So these, this is one of the varieties which is important. So nowadays, how we have, reduced, as I have already told that, 7 to 10 generation or 11 or 12 generation sometimes to, back cross, and then you will get the trait integrated into the recipient variety. So the important thing is that how this has been got changed in the recent era. So that I'm going to discuss in detail. But before that, first I will discuss about the impact of Green Revolution. So what is the impact of Green Revolution when they have started with the agriculture, with the yield of the mainly food crop of rice and wheat? Besides this rice and wheat, other crop variety also got, they have adopted this technology, and they have, done the breeding program. So many other varieties. So, and then a nation attained self-sufficiency in the food production. Now, that time, the FCI has people, the government has started maintaining buffer stocks in FCI, which has been established in 1964 for the food, fertilizers, and seeds. And then the government was able to curb the inflationary trend of the food products. So these are some of the impact of the Green Revolution. These are the basically positive impact. But what I am going to discuss is also the negative impact, mainly the environmental impact for the Green Revolution. So what has happened? You just see this particular slide of mine. So where you can just see that in a 1956, 55, 56, just that you can just see that the pesticides consumption over the years in India is just the very less when in 1955, 56. But you can just see that this is the time, say 73, 74, 88, 89, 91, 92. These are the time when the green revolution has was in the peak. So you can just see the per capita consumption of these the pesticides has increased. Although in latter years, when people have got the environmental impact or the harmful effect of these pesticides in human being, so then they have started growing the plants in a different manner. So it has gone down, and you can just see that it still is being maintained. It has never come to the point where we were there in 1955, 56. So how this has affected in our the country, that you can just see that the per hectare fertilizer used by the states in India, this is the data of the entire India, but you can just see that which one is the highest. Punjab, which is known for the start of the green revolution in our country, and later on the AP, Tamil Nadu, Haryana, Bihar, UP, everywhere, all these people, even the Karnataka is also higher than the Indian the average. So what is the bad effect of these things has happened. Like you can just see, you can just Google cancer train in your mobile phone, and you can find all these pictures in your Google search. So there, this is a really a shocking tale of India's cancer train. This was, the if you in the this is the picture that I have downloaded from internet. So this is, basically there was a place is called Bhatinda. From there, there was one overnight train used to start in the night, and it used to reach to Bikaner, which was in Rajasthan, in the morning. Bikaner was having a cancer hospital, and then near about the entire the train was full of the cancer patients. Near about 200 to 400 patients every day they used to travel along with their attendants. So that's why they have regarded or they have named these things as a cancer train. So this is the one of the important place where the indiscriminate use of these pesticides and fungicides has actually given a very bad impact on the human being as well as environment, as well as even the fertilizer. You must be if you have a village background, if you go to your villages, you will find that most of the villages ponds are now turning green in color, or because of the algal growth. And this is because of the fertilizers or chemical fertilizers which we are using in our field, which is getting leached out from the field, and it is actually being collected in the ponds, and it actually promotes the algal growth in the ponds. So this is these are some of the challenges. So when we want to do what we have learned from our past, that the first green revolution was, even though it has given a self sufficiency for the agriculture food production, but it was not a sustainable growth, that what I feel. So what that the challenges what we have in for the green revolution in 21st century is that saturation of the crop improvement opportunities, that already we are feeling a lot. Like in breeding program, if you can cross a certain variety of, suppose it's a wheat variety, you can cross only the certain varieties of the wheat. Mainly the some of the resistance against the different pathogens, that those locus or gene is basically present in many of the crop plants, in the wild, their wild relatives. So what has happened, like if if we want to cross them, they usually don't get crossed. Besides that, you can cross like wheat to wheat, but if some good gene is there in the tomato, and if you want to integrate into the wheat, that you cannot do. So that's why there is a saturation of the cross improvement opportunity what we are feeling. Second thing is that what has happened, like after 4 5 generation, the plants are growing in the field. It happens like after sometimes the again plant becomes susceptible to those pathogens, and some new diseases started coming. So all these things started the thinking that we should have certain there's a long existing problem, and we should have we can increase our crop productivity with a sustainable growth, and with less impact of the on the environment. That the sustainable agriculture and environmental perspective is there, and the speed of improving crop is for better yield and value addition. That is also one of the important challenges of the 21st century. So if you see this per capita availability of the food grains in India, so that you can just see that in 1951 it was the near about 120 24 gram per day per person. So now if you see that in the case of now we have reached in 2024. So it's that data which is available in 2021 is near about 342. However, the if you compare to the international standard, still we are very very less. So our population is increasing, and in geometric ratio, and the food production in arithmetic ratio. So there is the gap that we have to fill, and how we will fill with the new generation things. So there is a now the need of second green revolution, what we call. Okay, so this second green revolution term has been, given by our Prime Minister. He has called, the second green revolution to be started. I think in 2015 that he has given this thing, and then the major challenge is that how do we feed more population without further damaging our planet. So that means the sustainable the research is required for the development of various traits and the new characters in the plant, so that we can have more productivity, and that will be useful for the feeding of the masses. So the government has started the some of the things which we call as a soil health card. So and these are important, like you can get your soil tested, and then you according to the, suppose there is a, usually in the fertilizer, we either go for nitrogen, phosphorus, or sulfur. So potassium, sorry, potassium. So NPK is there. So you based upon the requirement of the different fertilizers that you can apply. So basically, the second revolution, how we can do, and what the important challenges are, basically have we learned from our mistakes of the first. Then GM plants or genome edited plants will be helpful or not, that we will discuss in today's lecture. And the myths and apprehensions about the GMOs, the public perceptions and acceptance of the new technologies, and the way forward that we will discuss in the end. So GM crops and genome edited crops, the major apprehensions usually people do have, that they are different from naturally occurring plants, which there are so many logics and things are there, which and the experimental evidences that what we eat right now is already modified several times by our forefathers. Is say basically what we do have, what we are eating is actually the it is not the same crop which people have started growing near about 10,000 years back. So how they have got that is called as a selection breeding. that they all have done in this way. That you also must have realized, that if you go to villages, you will find that the most of the farmers, they keep the seeds for the next generation, and whenever they require to keep the seeds for next generation, usually they take a healthy and good, character seeds for the next generation. So this is how people have started, the long back, this selection breeding, and now it is unknowingly or unknowingly they have, selected our plants, which is already, having so much of, variations in the field. So they are going to change the another apprehension is that they are going to change the entire gene pool. They are bad for the human and animal health, and they are bad for the environment. We will discuss about all these points. So basically, the myths of the natural food is that the food that we eat comes from the plants already extensively modified from the original form. Even the heritage varieties are extensively genetically modified. So this, if we talk about the distant past, near about 10,000 CS back, so our forefathers were, they were started, the most of the, the what has happened, like the when people were actually nomads, and from nomads to, when they have started the society, it's basically most of the societies or all these things has been, developed in the river banks, because of the, the why they have, all these, they have developed in the river bank, because they were actually found some of the lands for the agriculture, and for the agriculture you have to sow the seeds, you have to wait for the crop to get harvested. So they then they got aggregated from the nomads to, the societies, and so the most of the civilization has been developed in all these points. And you can just, these are the some of the evidences, although, there are some evidences of rice and bean in India near about 8,500 years ago. Rice, soybean is near 9,000 years ago. It's not that only these two plants has got, evolved here, but we don't have the evidences for other, the crops. So, but for the wheat, barley, this is corn, squash, beans, potato, near about 10,000 years ago. So all these things started, long back by our forefathers. This is the selection, how the people used to do this selection breeding. You can just see that this is a maize plant, so if you see that there will be a natural variation within the population. So out of these, the different crops, which crops that we are going to select is the healthy, the higher, better seed quality, everything should be all right. So we are going to discard the varieties which are, low yielding, only the plants which are which may have developed by the natural variations, the better for the next generation, for next generation. Likewise, the people have got these things. So there are some evidences in Mexico where the people have found the crops from the archaeological sites of the valleys of Mexico. You can just see that it was there, near about 7,000 years back. It was a small one, but you can just see the size of the increasing size of the cob is there. So that means, why I mean to say that all these things, because now people believe that we are going to change our naturally occurring variety, but all these have already, being, selected long back. And important thing is that, the new varieties which has been developed, where, the earlier varieties were, the maize varieties were actually they were branched, and the seeds were also having a hull which was protecting the harvesting, for the harvesting process. So it was difficult to harvest. So then people have selected the open type of seeds, as well as the single, now if you see the maize plant, it is a single erect plant, not the branched one. So that's how people have started selecting all these things long back. And whatever the if you see that the breeding program, this IS-64 rice has been used for the this breeding program during the Green Revolution, first Green Revolution. And you can just see that origin rice, and some of the Mexican variety, when they have done all these manipulations, and the IS-64 is now already having manipulated so many times. So the nothing is natural. So that I mean to say by all these things. So what are the different advances in the genetic technology which can contribute to the improved plants? One first thing is the marker assisted selection. Second is the we talk about the speed breeding, that I will discuss a little bit, and the marker assisted selection also I will discuss. Then recombinant DNA technology for the transgenic plants, and the precision genome editing, that which is recent things which has been added in by the plant biotechnologies. So what, if you remember my earlier slides, when people used to do the classical breeding program, where you have to wait for at least the different generations to back crossing against to get homozygous and heterozygous plants, and ultimately you are going to have at least 7 to 10 generation for the breeding programs. But marker assisted selection, where you are using molecular markers for breeding programs, that has actually reduced everything to 2 or 3 generations maximum, sometimes 4 generation. And there you can get the homozygous heterozygous plants very easily. So there, if you see that this is a susceptible plant, this is a resistant plant against a certain pathogen, and you can just see that susceptible plant is having, in the gel picture, you can just see that the band is here, and the system plant, the band is here, by using certain markers. So what will happen in the case of the next generation? Here you can just see that this is, you can just figure out these are the heterozygous, these all are heterozygous, but this is the resistant plant what we have got. So resistant plant you can get here, these two will be the resistant plant, which is the homozygous to each other. So all these things that you can get by this breeding. So this is easier to find out even in the F2 generation, that which of the plants are important for your further programs, and you can select the programs in the first generation, I mean the F2 generation only, which was earlier, people used to depend upon the phenotypic selection. So if there is a susceptible plant or resistant plant, so they used to spray the fungal spores or bacterial spores, the pathogen the on the leaves, and then they have to wait for the different generation to get the data for the breeding programs. But now you can in F2 generation only, you can figure out that which of them are for either resistant type or susceptible types, as well as the homozygous and heterozygous, so that you can use for them for the further studies. So nowadays, the not only that this marker assisted selection breeding is for one character, now people are using for gene pyramiding. What we used to do is that 2 or 3 different characters we used to combine together by these marker assisted breeding, and this you can select very easily in the different generations. So now the time has got reduced because of this marker assisted breeding. Second thing is that because of the marker assisted breeding, the we can do integration of certain characters in more number of varieties. Suppose if the variety which is local to the Karnataka, and one variety is local to the Madhya Pradesh. So what we can we can suppose if you have developed a variety in Madhya Pradesh, that may not be working very good in the Karnataka variety. So it's always better to integrate all these genes, so in different varieties, This has actually increased the speed of the breeding programs. So marker assisted selection breeding is now is important. Where here you can just see that you are just going to take those plants which are resistant in nature for the marker assisted selection. So all the in the with the evidence of genome sequencing, now we are getting by different methods are there like GWAS and the QTL-seq, many other methods that we are using right now in my laboratory also, where we are utilizing all these things in details, and is important for getting a new variety very easily by these marker assisted selection breeding. So the most important thing is that the this marker assisted selection breeding, that you don't have to wait for the phenotype. Suppose if you have just one plant and then you are using the pathogen and plant will be dead, so then how you are going to screen those plants for the next generation? That plant will be dead, or it will be some of the. And most important thing is that sometimes when you want to do this type of breeding program, you can you have to always depend upon the field data. So usually the plants which are used in the greenhouses or glass houses, they don't give the same result which we obtained in the field programs. So but this marker assisted selection is now is important for not only for one gene, for many genes that you can integrate in the plants in a very fast and very sustainable manner. So this is the benefits of the that more accurate and efficient selection of the specific genotypes. They may lead to the accelerated variety development, that is important, and also that they are more efficient use of the resources that you can do, and especially for the field trials that you can do very easily. So like here in the markers genotype, you don't have to do much of the experiments. You just take the leaf or sample. You don't have to take entire plant, just a small sample. Go for the DNA extraction, PCR, gel electrophoresis, and then you go for the marker analysis. Such type of things that you can do very easily in the markers selection. And then you go for the molecular breeding of this is my lab data that we are right now doing in the chickpea variety, where we are trying to interrogate the multiple fungal disease resistance. I've got a big project on this, and where we are with the help of IRI, Indian Agriculture Research Institute, we are doing the interrogation of two diseases. One is the Escocata blight, and another is Fusionum wilt, to in a same plant. That we are doing back crossing and then again crossing. So this is something we could do, the gene pyramiding. You can just see that he's my collaborator in IRA, a very known person in chickpea breeding, Dr. Bharadwaj. And you can just see that these varieties are resistant varieties, and the variety where we have the lane where we are standing is actually the susceptible variety which has all vanished. This is a sick plot of the fusarium. So these all are vanished in this particular field. So this type of things people have started in many plants, and they are getting very good results in this. So then comes in this new technology which is called as a speed breeding. So this speed breeding is mainly, what is speed breeding? You can just the different light people have started using. So this technique has been developed in Australia, Western University of Western Australia, and now it is being adopted everywhere in the world. So what the suppose if you want to get one or two you have to wait for the season. Usually even in greenhouses you cannot do breeding for more than 2 or 3 times in a year. So those things, like it will take time for all these things. But by speed building technology, where they have reduced the darkness period, that is 22 hours is the photo period for the light and 2 hours for the darkness. And this is the different light qualities and everything, the humidity and temperatures is basically speed building. You have to set up a different type of greenhouse which is suitable for the speed breeding. So now many places that government of India has given money to get it established. So there, the important thing is that you can do 8 to 10 generations sometimes. With the people have shown in a year time that you can get the seeds of for by this speed breeding. So the 8 to 7 generation that you can get achieve. So sometimes people have also achieved with the chickpea. Also, people have achieved 6 to 7 generations. Wheat near about 8 generation, and some of the other variety they have generated more than almost 10 generations in a year that they have got. So this is the advantage of this speed breeding, where the generation time is important. And here, the light cycle is that it is more compared to the dark cycle. And there are some methodologies there by which you can do this speed breeding. And the speed breeding is accelerating the generation time of major crop plants for the research and breeding. So then comes the GM crops, and usually what happens, like there, in the breeding programs, you can either cross between the tomato to tomato, potato to potato. You can cross wheat to wheat, rice to rice. But suppose some disease-resistant gene which is present in the Arabidopsis thaliana, that is not same of the elite variety, but that is important. So what you can do is you can just excise this disease resistance gene, and then you can transform into the tomato, which is a elite variety, and then you can get a tomato which is resistant against a certain disease. So this is the advantage of the GM crops, where you are going to have, you isolate, you manipulate in the test tubes, and then you can go for the transgenic plants. There are a few other techniques are there also, what we call is cisgenics. Cisgenic is basically, suppose there is a gene from apple, that is a red apple, which we are going to transform into the one apple variety which is green in color. That is called as cisgenics. When it is from Arabidopsis to apple, it will be transgenic. So cisgenic is also very important. Like suppose I got a very good gene which is present in the variety of chickpea, which is not crossable to the elite variety, but I can isolate the gene and can transform into chickpea. So there should not be any problem in when I am taking a gene from the same plant of the different character, and I'm just inserting into the same plants. So this is how you can do this cisgenics and transgenics plants. So people have already done several this type of, these are examples. One thing is, when we talk about the BT, the GM crops, always, the BT comes in our minds. BT is basically isolated from a Bacillus species, and after this Bacillus species, people have found that it is actually it is toxic to a particular type of the insect larva, Lepidopteran larva, and it is actually going to kill. So that that when they have transformed, they got the disease resistant plant. So if you see that, the most of the time the people who are against this GM crops, they are actually most of the time they are citing, the BT example where the bacterial gene is being, transferred to the plant. But what about cisgenics? When, the plant, the same variety, some gene is, we are isolating and, transferring to the same, the genus and species, but a different variety with the different characters. So the BT is basically, that like it actually is a protoxin, and when it actually interact with the pore assembly of a particular lepidopteran insect gut, so then only it will become toxic. So it will convert it to toxin, and then there will be pore assembly will be there, which is important for the absorption of the nutrients in the gut. So that then slowly the insect will be dead. So what the my experience is that I have traveled throughout India, and I found the mostly the if you go to Maharashtra or Gujarat or even the Telangana and Andhra Pradesh region, so there you'll find that the cotton farmers are almost adopted this near about 80 to 90% of their fields are basically BT cotton they are growing. So they all these things is happening, but important thing is that still, we are not accepting all these facts. There are so many reasons are there. One reason, most important reason, is the political reason. So that as a scientist, I will not comment on those political reasons, but you all understand what are the political reasons where the people are actually against the MNCs, and they think that all these transgenic plants, when it will be developed by MNCs, and they are going to stop they are going to capture our farming things, and it will be important for all of us to sustain in our own niche. So what has happened, that like it's not true that only the MNCs, the Monsanto and other Syngenta, all these are people are actually developing this transgenic plant. But what about the universities, institutes? They are also developing. They are not doing for profit. So there are so many arguments are there. So we will discuss in the latter part of my lecture. So there are the beta carotene, which you all know about the golden rice, which has been developed. So these are golden cauliflower, golden maize, all those things are people have already done. This is one of the example of the GM examples of the disease resistant banana. This was, actually in the Congo, there was, or GRA. So one country is there where, in the Eastern Africa, what people have, got a gene from the chili, which is resistant to the for the bacterial wilt disease. So they transformed to the banana plant. Banana was most economic plant of these. These are poor countries of Eastern Africa where they have developed this transgenic plant, and they have given to those things. The one of the Indian scientists, Dr. Leena, is involved in that, and she has got the highest civilian awards of those countries, because they were almost in the on the verge of famine, and and now they have got very good resistant plant, and now their economy is increasing. So such type of things are there, in India also this is one of the article that I have captured from internet, and that has appeared in 2017, where genetically modified crops where that they have started this by in Delhi University, where Professor Pintel, he has developed some of the variety of by transgenic of this brassica Johnsia, and there are so many things. It was almost about to release, but there were some problems, and they again the anti-GM people, they have started talking about all these things. I don't want to go into details. You all know about all these things. There are many people who are against with this GM crops, but there are some the farmers, they are actually trying to in this is the Shetkari Sangatana, which is in the Akola district, where they have gone for a march for in the favor of GM crops. There are in the if you talk about like in Bangladesh, the many of the brinjal variety, they are actually being released by or approved by the government. They are actually growing. So somehow some of the seed has come from the Bangladesh to India, and people are the people have found that it is being grown in Bihar and Bengal of our country. And there it's very difficult to figure out all these things in whenever it is not there. So what has happened, like it is always important to have a good system where we can have checks and balances, and then we can release. Otherwise, people will start adopting all this technology in advance. So there is another technology. So some good part of which has happened in recent times by government. So that is the genome editing. So usually, the targeted intervention of at molecular level of DNA, which is deliberately to alter the structural and functional characteristics of organism. That is genome editing. This has been recently discovered, and this is called as site-directed nucleases. So there are different that I will discuss about those things. SDN1, SDN2, SDN3, different type of site-directed nucleases are there, where you can have the zinc finger, talon, and CRISPR-Cas. CRISPR-Cas is the recent one, and it is being widely used. Government of India has launched so many projects and given funding to so many projects which are now being carried out in various institutes and universities on CRISPR-Cas system, where you can do the precise genome editing. In the transgenic plants, the it was not that precise, because when you are transferring to the plant, it may be going anywhere in the genome. But here you can precisely in the genome itself, you can cut, and you can repair those the plants. So the plant breeding has made enormous contribution to increase the global food production, but there are limits. So basically the GMOs and these plants are the genome edited plants are new hopes. So process is basically the plant breeding is that the precise and in inserting the desired trait, fast to obtain the final product, and can increase the crop productivity and quality, producing the resistant plant, biofortification, all those we can do. But cons was for the GM crops was basically the random nature of the gene insertion, not favorable to making large concerted changes by adding the entire metabolic pathway. Only one or two that you can change the genes, and it's heavily regulated now. People have found. So then now the our basically just wait. So basically, it's a now it's a what we call as a new era where the genome editing is important for breeding the plants in a very precise manner. So these are basically site-directed modification for the new products, reduce time to get the product, and reduce the number of plants which are involved, and also introduce the longer DNA sequences into the plants. So these are some of the important things that which you can get in the genome edited plants. So there the applications are research is one of the things that people are right now doing to elucidate the gene function, that if you want to mutate a gene, you can use this genome editing very easily, and this is fast. This eliminate these undesirable products that negatively impact on food quality, storage, and processing, accumulate the metabolites of the values like fatty acids, and then simultaneously create mutations of multiple gene family members, and they create the varieties resistant to pest and diseases, they among many others. So what has happened, like recently government of India has approved that the exemption of these genome edited plant falling under the category SDN1, standard-acted nucleated one, that is that just precise editing, and the SDN2 of under the provision of the rule, that is 1989. So basically, the rule, there is a bill is also pending against these the transgenic crops and other manipulations. So it is lying in the parliament since 2003. So I don't know whether how it will be going to be passed, but it will be really important for all of us to pass a rule. Let's bring all these things in an umbrella of law, rather than people are working on different aspect, and they are releasing the varieties in the field without any checks and balances. So it's better to have a law, working under law, that will be better. So there are, the government of India has also, the Ministry of Consumer Affairs has already, given this things in January 1, 2013, that every package should be labeled whether it's a GMO or non-GMO. So this is, some of the development that which we do have. We do have, the agencies as per the 1989 law, and there it is being conducted by two ministries, basically for the release of transgenic plants. One is by Ministry of the Environment, another by Department of Biotechnology. Here the Ministry of Environment is having the GEAC, and RCGM is the another, the body is there, which is actually looking after these things. And there are so many things are there, usually the people who have developed some of the transgenic plant which may be useful for the farmers, actually they stuck in these the ministries most of the times. But nowadays, the what we have realized that is thinking is being changed a little bit. Government is, so many ministers are also supporting right now. Even our prime minister is supporting, but without law we cannot do anything about all these things. So there is a, I'm just ending my talk with a positive note, that if we link public policy, science, and public understanding for the bright future in India, we can make a difference. So this is with this positive note. Thank you very much. I can interact with all of you if you have any questions. Any, this is a general type of lecture, so you can ask any question. If I will be able to answer, I will answer. So this is the aerial view of my university, Jawaharlal Nehru University. You must have heard about this university in a bad name, but this is a beautiful university. It's the #1 university in India, and the campus, you can see that it's a beautiful campus. My lab is somewhere here in this particular building, and this is our central library. Thank you very much, and you can write to me on this email ID. Thank you very much. You can. Over to you, Professor Rajkumar.
Good morning, sir.
Yeah.
Am I audible?
Yeah, sure.
So